Perfect. Can you see my slide?
Yeah.
Excellent.
So today we're joined by Dr. Omarjee, who's going to talk to us about his career and immune response. And over to you, Ommar.
Well, thank you, Cindy. So hi, everyone. I'm an immunologist working at the University of Cambridge. So basically, my work is to try to understand how does our immune system works. And I'm sure like within the past 2 years, you have learned a lot about immune system with the pandemic we've had, about viruses and vaccines. So today I'm going to talk a little bit about our immune response, so how our body provides an immune response, and we are going to have a closer look to a specific type of immune cells called CD8 T cells. During this presentation, I will talk about different things. So like I said, I will describe what is an immune response. Then we'll focus on T cells. Afterwards, I will take you inside of the cells to see what are the different components that make a T cell function. Then we'll move, I'll give you a practical view of the type of experiments that I do. And finally, I'll talk about my education and how I came here to Cambridge. So let's start first with an immune response. So I will start with a question for you. So it's an open question. Can you tell me what is the role of the immune system? So Cindy is going to, so you can answer that in the chat, if I'm correct, Cindy. Yeah. And we will have like 1 or 2 minutes to give me a few answers.
Okay, so does anyone know what is the role of the immune system? If you can put what you think in the chat box, and we'll give you a few minutes for that. What is the role of the immune system?
So don't be shy. So I'll help you over here. If oh, great.
Oh, we've got some answers. So we've got to protect the body and fight off disease. Very good. And it allows us to defend against pathogens.
Great, absolutely right. So our immune system acts as a shield to protect us from external threats, so which are like contamination with bacteria or infection with viruses such as SARS-CoV-2, which generates COVID-19 disease. But it also helps us to, it protects us against internal threats, for example, which are cancer cells, which are cells that originate from our own body. And in order to be able to give a good immune response, we need different types of immune cells. So another question for you, can anybody know, does anyone know some immune cells?
So again, if you want to just put your answers in the chat box. So does anyone know some immune cells? I'll give a couple more minutes for that. Ooh, memory cells, macrophages. Yeah.
Yes, macrophages is a good answer. Does anybody knows more immune cells?
We've got T and B lymphocytes.
Yeah, correct. Yeah, and maybe one more, and before I give you the answers. Maybe not.
Yeah, I think that's all the answers we've got for
Yeah.
this question.
Perfect. So, yes, you were right. So macrophages, T and B cells. So basically, our immune system has an arsenal of different immune cells. So we said macrophages here. Someone said T cells. So there are different types of T cells. We call CD4 T cells, CD8 T cells, B cells or B lymphocytes. And then also different types of cells called monocyte, basophil, eosinophil, neutrophils, and dendritic cells. But all these cells can be divided in two main groups called innate, so which belongs to the innate immunity or the adaptive immunity. The innate immunity is our first line of defense. It will provide a fast immune response, but at the same time, it is short-lasting. Whereas our adaptive immunity is slower. So as the name suggests, it will create an immune response which adapts to the specific pathogen that the body encounters, but will provide a long-lasting response. So we are talking about the concept of memory. And this is basically how vaccines work, because vaccines will activate the adaptive immunity to be able to provide us with a long-term defense against pathogen. Now, one last question for these slides for you. Do you think an immune response is always good? Yes or no?
So again, just in the chat box, do you think it's good? Yes, or always good, should I say, sorry. Yes or no. So we'll give that a minute. Okay, so we've got no with an explanation. So we've got no because, for example, some treatments can cause the immune to react and possibly reject the treatment. And we've got another one saying it may be harmful if an immune response occurs against healthy somatic cells.
Yeah, absolutely. I'm super impressed. That's great answers.
And not always, as there are autoimmune diseases.
Yeah, autoimmune disease, very nice.
Fantastic, yeah.
Cool. So yes, you're right. So first of all, there is the normal immune response. So when the body encounters a pathogen, it will mount an immune response. So the intensity will increase, fight the disease. And once the pathogen is eliminated, the immune system will go back to normal. So all the immune cells will die or will be eliminated so that the immune response goes back to zero. But there are cases, for example, where the immune system, the response is too low, is not enough efficient to be able to fight the pathogen. So we talk about, in this case, we talk about immunodeficiencies. So an immune system which is deficient. And in this case, we can have repeated infection or even pathogen, which in cases is not harmful for the body, but in this situation will be harmful for the patient. On the other side, you can have an immune response which is too high. So you might think that, oh, if it's too high, so it will eliminate the pathogen rapidly and everybody will be happy. But actually no, because a too high immune response can damage the surrounding tissues, and also in the absence of pathogen can recognize our own body as a pathogen and start attacking our body. So in this case, we are talking about autoimmune disease or autoinflammatory disease. And an example of that is lupus or arthritis. So just as a side note here, so when I was doing my PhD, we were following families that had autoimmune and autoinflammatory disease, mainly those two, where, for example, the grandfather was sick, the parent was sick, and the children were sick. So there was a genetic aspect to those diseases that we were trying to understand what was the cause of that disease. So very good answers. Thank you very much for your participation. Now, within this arsenal of different cells, I am currently working on CD8 T cells. So what is a CD8 T cells? It is basically a cell that can kill infected or modified cells. For example, it is very effective against infected cells where pathogens are hiding inside the cell, for example, viruses or intracellular bacteria. And it can also fight against hosts, so our own body cells that has become different, that has changed from their normal route, for example, cancer cells. Now, in order to fight those cells, the CD8 T cells has two abilities. The first one is to be able to multiply, so you have more of them to fight their infected cells. And they have an ability to kill those cells by generating molecular bullets, if you call it like this, that are of three categories that we call perforin, granzymes, and cytokines. So the perforin is going to create, as the name suggests, is going to create holes into the cells, and the granzyme is going to enter through those holes in order to induce cell death. And the cytokines is also going to induce cell death through other mechanisms. So next, I'm going to show you it shouldn't start right now. So I'm going to show you a video. So it's a live video of how a T cells, which it will be represented in green. So I thought you might be able to see it now, but I'm just going to play it in a minute. So the T cells will be represented in green. So it's colored in green. And there is 3 tumor cells, which is in blue. And in that little square, we have a molecule called annexin 5 that is going to stain in red cells that are dying. So we will see how those T cells are able to physically touch the cancer cell, induce cell death, detach from it, and then go and hunt another tumor cells. So if you're ready, I'm going to play the video. So you have the T cell here, it touches the cancer cell, kills it. So it turns red. Now it waits, it jumps on the another cancer cell, induce cell death, jumps on the other one, and induce cell death again.
Just amazing.
So maybe we can replay that video so you can have a second look at that. Yep.
Makes me think of Flubber. When Flubber jumped around a lot. It's just so cool.
So this is very cool because this shows that the CD8 T cells is able to, through molecular signal, is able to know where the cancer cell is located and will jump on it, make a physical contact, throw those bullets, fire those bullets on the cancer cell, induce cell death, and then detaches from it and go and find the next target. Next slide. Perfect. But you know, the cancer cell is not going to let the T cell win either. So it has developed mechanism. For example, it can start secreting molecules, which are called immunosuppressive molecules, that is going to fight back. And those molecules are going to tire the T cells and induce a state of chronic fatigue that we call exhaustion. So in this situation, the T cells is no longer able to fire those bullets and is not able to fight against those cancer cells. And actually right now in the pharmacological or biotechnological industry, there is a lot of effort that is going around trying to see how can we limit this process of exhaustion or even reverse this process of exhaustion in order to keep a healthy population of T cells that can keep fighting against tumor cells. So now this is a quiz for you. Based on what you've seen, there is a multiple choice question that you can choose, and maybe Cindy can explain this better than me.
Yeah, so our first question is, How does a CD8 T cell fight an infected cell? So for this one, you can select a few of the answers. Then our second question is, how can a cancer cell overpower a CD8 T cell? And that's single choice, so it's only one of the options. So if you want to pick your answers, and we'll just give it a few minutes, and then I'll post them. I know the answers and I'm stuck. So just one more. Okay. So let's share results. Can you see the results?
Yeah.
Awesome. So we've got mainly by secreting perforin and granzymes and by inducing cell death. Is that correct?
That's perfectly correct.
Well done.
Yeah.
For our second one, they've gone with the first one by secreting immunosuppressive molecules.
Absolutely correct again.
Fantastic. Well done, everyone. I didn't know.
Amazing. So basically, currently, my line of research is trying to understand, trying to go inside of the CD8 T cells and try to understand how can we make them stronger. So I'm going to take you inside of a cell now and try to see what's in there. Now, so this is a cell that, this is a 3D picture of a cell. And there is a little task for you here so I've numbered different components of that cells by 1, 2, 3, 4. And basically, it's a little game trying to guess what are the different numbers here. So do you want to go ahead, Cindy, and explain the poll?
Yeah, so the poll is now live. So if you've got all your options, so cell membrane, nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, and cytotoxic vesicles. So if you want to select one of the answers, and we'll wait a few minutes for their. You've got all your numbers. So like, is a cell name number 1, 4, or 6? And same goes from below. We'll give it a few minutes. Got one response so far. So can you identify the different parts of the cell? I'm just checking if I know. The mitochondria. Okay. Endoplasmic reticulum. Okay, we have all of our answers in. So can you tell us if cell membrane is number 1?
Yes.
Yeah. Oh, brilliant. Everyone got that one right. So nucleus, is that number 2?
Exactly.
Mitochondria, is that number 3?
Perfect.
Endoplasmic reticulum, is that number 4?
True.
Golgi apparatus, is that number 5?
Yes.
And cytotoxic vesicles, is that number 6?
Wow. Amazing.
Yeah, everyone's done fantastic.
Cool. Excellent. So yes, you're absolutely right. So this is what you said. So number 1 is cell or plasma membrane. The second one is nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, and cytotoxic vesicle. And all these, everything that is inside the cells, like these components, we call them organelles, which also means like mini organ. So you can imagine the cells to be another kind of complexity where you have different organs that work together to make a cell function properly. And here I've mentioned it's a CD8 T cells, but you can keep in mind also that all the cells in our body has kind of a similar structure. These are the main components of a cell. And so just to give you a brief description of the function of those different compartments. So basically the cell membrane is the physical barrier that is going to contain the intracellular component of the cell. The nucleus contains the DNA, our genetic information, and is important for producing proteins. The mitochondria is the powerhouse of the cell, is going to generate energy. The endoplasmic reticulum has a lot of important function, and one of the mostly known function is producing proteins from signal it receives from the nucleus. The Golgi apparatus is another component that is going to mature those proteins into their final forms. And cytotoxic vesicle, in the case of a CD8 T cells, is basically vesicle that will contain the perforin and granzyme. And so another question, so it's an open question now for you. Which organelles here between 2 to 6 do you think I work on?
We have that as the poll as well. So if it's a multiple choice one, so it's between numbers 2 and 6. Which ones do you think Ommar works with? Okay, so we've got all of our results in. Let me share the results. So we've got, do you work with cytotoxic vesicles?
Not directly.
Do you work with the nucleus?
No.
Do you work with the mitochondria?
Yes.
And do you work with the endoplasmic reticulum?
Yes.
And the Golgi apparatus?
Not really.
So some got, say, 3 people got that somewhat right. So well done.
Perfect. So basically I work here with the mitochondria and the endoplasmic reticulum. And basically I try to study the interaction between those two organelles. Now, why do I work on those two organelles precisely?
We've got a question from one of our participants though. So it's just asking if any animal or plant cell is always like this.
Sorry, can you repeat the question, please?
Is any animal or plant cell always like this?
Yes, so actually there is a lot of components. So yes, like a lot of animal cells has, like most animal cells has the same composition, except for the cytotoxic vesicle. They will have other types of vesicle, but not a vesicle that contain perforin and granzyme. But otherwise, all these components here is true in all animal cells. If you want me to go in slightly more details, the only exception in our body is red blood cells that doesn't have a nucleus in order to maximize their ability to carry oxygen around. And also plant cells are slightly different also, where they have a stronger structure around the cell membrane called a cell wall. And they have another organelle called chloroplast, which has the ability to capture light in order to produce food. So but basically they have very similar structure.
Yeah, thank you.
Okay, so now I was going to say, so why do I focus on the ER and mitochondria? So what I've did here, I decided to zoom in on that section so we can have a look of the ER and the mitochondria in a 2D picture. So this is the ER, so which is like a membrane. So outside you can consider you have the cytoplasm, so basically this part here, and this is the internal space of the ER, and this is the mitochondria. So one of the first role of the ER is to be able to produce proteins from signal it received from the nucleus. So that's the major role of the ER. Another major role is it acts as a signaling platform. So it will be able to sense if the cell is undergoing stress condition and then send the right message in order to counteract that stress. And another role of the ER is it is able to make physical contact with the mitochondria through the use of special proteins that is going to allow communication between those two organelles. And the mitochondria is the powerhouse of the cells. It is the organelle that is going to generate energy for the cells through here that I've depicted as a factory called the electron transport chain, also ETC. And it also will also mediate the use of certain metabolites, for example, glucose, or not glucose, but like other small metabolites, and also produce the different groups of metabolites that is going to impact how the cells will behave and how the cells can give a proper immune response. So, and my role is trying to understand how does this contact site here is important for the function of the CD8 T cells. So on my next slide, I'm going to give you a practical view. So what do I do in the lab exactly to be able to characterize how is this interaction here relevant for the function of the CD8 T cells? And if you have any question, don't hesitate to put it in the chat box. So let's have a practical view for that. So one of the, so I didn't, so I was thinking about that. Sorry, Cindy, I'm improvising slightly for this part because this can be a good question. So so I work with human samples. So I work on human CD8 T cells. Maybe you can write in the chat box from which organ do you think I get my immune cells? So maybe a 1 minute, time for you to put some answers. Where do you think I get from which compartment of the human body do you think that I get immune cells?
We'll wait a couple minutes for everyone just to put that into the chat box. I've also got a question from someone as well. What is the production of energy in a cell? Plan with chlorophyll and animal any numbers?
So, Jorge, are you asking about how, in terms of kilojoule, how much energy is produced? In that sense, I wouldn't really be able to. Yes.
Yes.
So in that sense, I wouldn't be able to give you a precise answer, but actually the cells but what I can tell you is that the cell is able to regulate how much energy it would need depending on in a given situation. For example, when the immune cells is under a stress condition, for example, an infection, it will have to activate the cells, and during that time, it will have to generate much more energy than without an infection. But I wouldn't be able to tell you exactly in terms of kilojoule how much energy it will generate.
Good question, though. And we've got a response to your, where do you think you might get your T cells? Do you get them from lymph nodes and or bone marrow?
Yeah, so actually the lymph node and the bone marrow is a source of, is a very good source, is a source for immune cells. We have a lot of immune cells which is present over there. But however, if you want to get it from humans, and the spleen also, is also an immune organ where you can get immune cells. But actually, when you think about it, if we are working on humans, in order to be able to get lymph node, bone marrow, and spleen, it would mean that you would need to do biopsies. You would need to do a very invasive surgery to be able to get those organs, so which makes it very difficult. And actually, one of the organ where you can get a lot of immune cells is the blood, where it's a mildly invasive technique and where you can get a lot of immune cells. So my experiment starts with getting blood from healthy donors that donate their blood for research purposes. And from these bloods, which contain red blood cells, white blood cells are the immune cells. So I need to process that blood in order to separate, to get the specific immune cells. So in order to do that, I will do what is called a fecal purification. So fecal is a very dense liquid where you can deposit the blood on top of that and use a centrifuge machine to spin those tubes. After spinning the tubes, you will have the separation of the blood in different components. So this is like a real image of after spinning the blood tubes. So here you will have the red blood cell compartment that went completely down. You have the fecal. Here, I don't know if you can see, there is a white ring here, which contains all the immune cells. And on top, you have the plasma, which is the liquid component of the blood. So in here, after centrifuging the tube, I collect here this ring of cells where I get a lot of immune cells. So in that ring of cells, there are different types of cells. There are like CD4 T cells, there are B cells, there are innate immunity cells like monocytes, and there is the CD8 T cells. So I need to find a way to be able to capture only the CD8 T cells. And for that, I use a technology that requires the use of antibodies. So antibodies are specific molecules that can bind to specific targets. And these antibodies, which here I call anti-CD8 antibody, which means that they can recognize the CD8 T cells, they have a metal bead on top of them. So you have a metal bead, the antibody, which can itself bind to the CD8 T cell. So imagine this is my mixture of immune cells, total immune cells. So I will add this antibody in it. That antibody will bind specifically to the CD8 T cells. And if I add a magnet to that, with the metal bead, will be attracted to the magnet, pulling aside the CD8 T cell, while the other immune cells won't be selected. And if I pour this tube, all the arrest immune cells will drain away, keeping only the CD8 T cell. So this is how I do what is called a positive selection of my CD8 T cell. And then when I collect my CD8 T cell, I want to study the impact of the contact sites in that T cell. So one of the ways in biology, if you want to study the role of a protein, is to remove it. You remove it and you see how does your control condition differ from the condition where you have removed that particular protein. And one of the ways to be able to do so, to be able to remove that protein, is to use CRISPR-Cas9 technology, which is a gene editing technology. So as you know, proteins are produced from genes. So if I remove that gene, I will also remove that protein. Now, how does CRISPR-Cas9 works? I won't go into detail, but what I can tell you here, imagine they are programmable DNA scissors. So you can program this complex of CRISPR-Cas9 that will go inside of your CD8 T cells, find the correct region that you have programmed it in the DNA, cut it to inactivate the gene that produces the protein that you're interested with. So this will inactivate your genes. And when you've done that, you can take your treated CD8 T cells, put them in a plate, put them with some specific media, Because remember, those cells was in the body before and had all the nutrients to be able to develop properly. Now you've taken them out of the body, so you should be able to give them a specific solution or supplement to be able to keep them alive for several days. Now, after a few days, once you're sure that the CRISPR has worked, you can analyze those immune cells. So in our field, when you analyze immune cells, we use the term of immunophenotyping, which means analyzing immune cells. Now there is an open question for you is, can you tell me what type of instrument do I use to analyze my immune cells or techniques? What type of instruments or techniques do I use to analyze my immune cells?
Yeah, so think about if you want to answer in the chat box, any sort of equipment you can think of in the lab that Ommar might use, or a process. And we'll give that a few minutes so that we have time for Q&A at the end as well. Electron microscopy?
Yeah, I do use that.
Anyone else with any ideas of techniques or instruments or any sort of equipment you might have seen in a lab? There are some really funny ones you work with, to be fair.
That's true.
Mm-hmm. Some reactives to use colors to show some differences.
True. Yeah, I do. I do use that too. And I use that with an instrument that I will mention. You will see, I will mention it.
So do you want to show us all of the lovely skills and techniques and funky things you work with?
So these are the different instruments that I use maybe on a weekly or daily basis. So let's start with the first one. So someone mentioned electron microscopy. Yes, I do use electron microscopy from time to time, which is a very powerful technique, gives you a very deep picture image of the cell. Confocal microscopy is another technique that is able to give you also a very precise picture of the cell, especially when you want to look inside and try to see how is the ER interacting with the mitochondria. And you can, with the confocal microscope, you can use colors that can differentiate the two compartments. Another technique that I use is a Western blot, which is basically a protein analyzing technique. I don't know if you can see here, it's a form of a gel where you can place different conditions in each column. So imagine you have CD8 T cells. You can treat those CD8 T cells with different drugs, for example, compound 1, compound 2, compound 3, compound 4, and put them into columns and then have a comparison of all the conditions side by side and know if your treatment has affected some levels of proteins in the different CD8 T cells. Now, Jorge mentioned using colors to be able to visualize some differences. So yes, so I do use that, and I use that with plate readers. So plate readers are great because you can read you can read different cells at the same time, and it gives you a lot of data within a very short period of time. Now, I would say most immunologists use their favorite techniques called flow cytometry. So flow cytometry is a technique that will analyze cell by cell, so each cell at a time. So you can use a tube, insert it in the flow cytometry machine, but it will take one cell at a time and analyze it. And when it analyzes the cell, it will give you different information. For example, the size of the cell, the complexity of the cells. And if you use some kind of colored marker, it can tell you whether or not your cell expresses that marker. Another technique, instrument that I use is called a seahorse a machine. Well, it's not very intuitive, but the seahorse basically is a metabolic technique. So it will help us to understand how metabolically active our cells is. And for that, for example, it helps you to know how much active is the mitochondria, because it's able to sense the consumption of oxygen, and which is one of the way that mitochondria works. And finally, it's not very exciting, but I do need to use a computer where I have all my softwares in order to analyze all the data that I receive from those different instruments here. So this part was on the science part. So if you have any question here, maybe you can ask it, because I will next move to my educational background.
So again, any questions, you can either put in the chat, or there's a questions and answers section at the bottom of your screen as well. But if, what is Western blot?
So actually, so thank you, Jorge. So a Western blot is a technique that will help you analyze proteins. So do you want some more details on that?
We've never seen anything like it before.
That's perfect. This is that I feel helpful now. So basically, it's a technique where you use a gel that has holes in it, and you will take all the protein that is in a particular groups of cells, and you can migrate it in that gel. And so basically, you have a separation of bigger proteins on the top and smaller proteins at the bottom. And you will have, and it helps you to separate all the proteins in a cell. And so what I didn't say is that afterwards, if you want to identify a specific proteins, you have to use antibodies, just like the separation technique that I presented before. So antibodies that is coupled with a marker. And so you will be able to identify your specific protein. So basic, but consider that a Western blot is a protein analyzing technique.
That's much simpler than I've ever heard it. So thank you. That makes much more sense to me now.
Yes, it is. Yes, absolutely. You're right. It is an electrolysis and electrophoresis technique.
So shall we move on to your career? And then if you have any questions regarding the science so far. Xavier?
No, is this a new technique? Is this a Spanish word?
Sorry. Okay, cool, awesome.
Gracias. So how did I reach here? So actually, I come from a very tiny island in the Indian Ocean called Mauritius, where I did my A-levels in 2019. So I did an A-levels where my main subjects was a science subject, so maths, physics, chemistry, and biology. And I really like all my science subjects, but I did have a preference for biology. So I decided to pursue my higher education in biology. And for this, I went to Lyon, which is a city in France, where I did a bachelor in life sciences. So the cool thing with this program was you started very broad. So with biology, and I could study different aspects of biology. And eventually, during the years of bachelor, I could slowly choose what subject I wanted to do and slowly specialize towards more genetics and cell biology. So here, and I'm also giving you some tips, but what I find useful for me. So if you feel that you resonate with them, you can go ahead and we can discuss about them if you want. So the first thing that I've learned during my career is that you need to choose something that you enjoy. There is no need to if you don't like something, maybe you need to rethink about what you want to do really. So choose something that you enjoy. And also during that bachelor period, I really wanted to get exposed to a lab environment. So during my summer holidays, it wasn't necessary, but I was contacting researchers that work in different labs, trying to see if they would have some space to welcome me so that I could work on their project and learn and start learning laboratory techniques. And during that time, I got to do 2 lab rotations during my last 2 years of bachelor. So during that time, I learned that you need to be proactive. So if I didn't go and look for those internships, I would never have been able to get this experience. So be proactive. And this is something that I'm still doing whenever I want something. I don't wait for the opportunity to present itself, but instead I create ways so that the opportunity comes to me. And then afterwards, I did a master's in genetics and cell biology. So it's like the 2 subjects that I really enjoy, and where I did 2 internships in immunology labs. So, but working on, so one lab was working on the adaptive immunity and the other lab was working in the innate immunity. So just to know which one I really wanted to do. And then following that, from these 2 internships, there was one lab that I really loved. And I decided to pursue a PhD in that lab, so that would specialize mostly in immunology. So basically the lab that was working on patients having autoimmune diseases. So during my time as a PhD, I learned that you need to ask a lot of questions. There is no need to be shy. Like when you don't know something, go ahead, ask questions. It will be beneficial in the long term. And also, I've also learned to be transparent about your mistakes, especially in the field of science. It's very important to be honest and to be transparent. So when you do something wrong, you admit it and you learn from it so that you don't repeat that mistake. And also, I've learned that you need to follow your instructions. Sometimes when it comes to a given experiment, you have this idea that you want to take it forward, go ahead and try to do that. And also I've learned the importance of collaboration and teamwork. And here I've used an African proverb, which I like a lot, which is alone we go faster, which is true, but together we go further. So, and collaboration is really nice. It helps you to grow. It helps you to interact with people and know how to be able to work in terms of a matrix organization with different groups of people. And so during that PhD, I've acquired a lot of technical skills, which is, for example, we talk about it, Western blots, flow cytometry, these kind of skills. But I've learned a lot of transferable skills, also known as soft skills. Now, one example of a transferable skills is time management, trying to know how to allocate your time to different aspects of the project. So now there is another open question for you is, can you give me some other example of transferable skills?
We've also got another question from Jorge. How old are you, Ommar?
I'm 31, but I do look younger, so I can cheat on that.
Yeah. So transferable skills, we'll give 1 minute. So what was your example of a transferable skill again, Ommar?
Time management, trying to know how to allocate your time to different aspects of a project.
Got language and cultural aspects.
Yeah.
It's quite transferable.
True.
Ooh, communication, teamwork, organization, adaptability. Some great answers as well.
Yeah, absolutely right. So yeah, excellent. So I'll give you some more examples. So I think Zuleika and Jorge, I gave a lot of examples. So during my time of a PhD, I was supervising undergraduate students and some medical students also. So that really taught me how to manage people on a project. Communication skills, so different types of skills. So for example, learning how to present scientific data to a community of scientists, but also to like the general public, a bit like what I'm doing right now. So you have to break down the scientific language, so it's very important. And then there is, for example, interpersonal skills, so how you interact with people than just more of a professional way, so how do you really connect with people. Critical thinking, because you're working on a scientific project, so you learn how to break down problem and how to think about what are the areas that you need to solve, to focus on. Problem solving, project management, all these aspects. And transferable skills mean that they are skills that you can take from one job and apply it to another job. So here, one advice that I would give you, that during your, like further down in your career, like when you start going to university or even now, try to think of ways where you can develop and refine your soft skills, because these are the most useful skills. I'm not saying that technical skills is not important, but betting on your transferable skills is also very, very important. And afterwards, after 2020, I moved to Cambridge, where I'm currently working as a postdoc. And here I'm leading more projects, I'm more independent, and I have much more responsibilities. So one thing I would say here, when I was younger, I thought that to be a scientist is to give 100% of your time to the lab. But actually, I've learned as from the PhD, it's not really true. Like, you will allocate maybe during the PhD 60–70% of your time to do like experiments, but the rest of the time is like you're reading a lot of scientific papers, you're doing presentations, so you're participating in lab meetings. So there are other aspects of your life, of your job, which does not require you being in the lab, but being like more in doing other type of activities. And being now a postdoc, I'm doing more of that, and my time is slowly decreasing. My time in the lab is slowly decreasing. And one thing that I've learned during the postdoc is that so there are different ways to choose a postdoc lab. For me, what was most important was trying to find a friendly lab and find a mentor, a supervisor that was going to support me and support my professional development. And finally, so this was my journey as a scientist. And one thing that I've learned as a scientist is that a good scientist for me is always learning. So thank you for your attention. This will be the end of my presentation. If you have any question, I'm very happy to answer them.
Thank you very much, Ommar. So we've got a couple. So a PhD can work in industry, and how? So I know we've got loads of PhDs, well, quite a few available at Wellcome Sanger Institute, but how would you recommend maybe getting into it? Or
yeah, well, I can expand this on this question a little bit more. So when you start doing a PhD itself, there are some partnerships that exist between the academia and the industry, where you can work, where you have one leg in the industry and one leg in your academic lab. So you can already get exposed to that environment. And once you are done, let's say you have done 100% academic PhD, and you're thinking of moving to the industry, well, there are positions of scientists where you can apply to them if your skill set matches to the position that they are advertising, or you can do a postdoc, what I'm doing right now, gain more skills, and then transition to the industry. But I would say that it is not relatively easy, but as long as you have the skill set that matches the job description, the job that is being advertised, for me, that there doesn't seem to be, there won't be any problem for you to be able to get access to those jobs.
So again, being very proactive about it and searching for what you want.
Yeah.
And we've got another question from Jorge about how the compromise with knowledge and research can work with commercialization.
Jorge, could you give us a little bit of more detail? What your what do you want to, what aspect of that question you want me to answer?
And if anyone else has any questions for our scientists before we end the session, if you want to put them either in the Q&A section or in the chat box, please. But this will also be available after the session, and we'll post it to It's Learning so you have more access to it.
Can I jump in with a question for you, Ommar? You've lived in sort of lots of different countries. I just wondered what your experience of that has been as a scientist, and what maybe you've gained from those different experiences.
Oh yeah, absolutely. So let's consider my jump from Mauritius to Lyon first. Well, to be honest, the first few years, not first, maybe the first year was slightly challenging, like to adapt to a new culture. But eventually, once you do that, you're exposed to a new way of working, of studying, of getting exposed to new types of science. So it's very, very enriching, I would say, if you have the opportunity to go abroad and to be exposed to a new culture of work. That's very enriching. I think there is no price. It's priceless. And so what I've noticed more specifically is that my personal experience in the French lab was that people like to have a very precise question and to work on that, and it's a very precise question. Whereas when I moved to Cambridge, I felt people was like more adventurous with the question, like they were able to like test more risky questions, you know. So I did feel that this was like, for example, two different mindset of how to approach science. But this can also be very subjective to the type of environment I was in. But I'm very grateful with both my supervisors that I had in Lyon that helped me during my PhD, and my current supervisor now in Cambridge. But I think that's the main, that for me is the main difference. So does that answer your question, Sam?
Yeah, no, I just wanted to sort of see what your takeaways were from your different experiences. But yeah, no, that's really interesting.
Yeah. So Jorge, coming back to you. So if I'm trying to understand, are you saying that from the work that we do in the academia, the research that we do, are we interested in selling that research? Is that what you're trying to say? Like, are we more interested in being able to patent or sell that research?
Maybe. I mean, it's one of the things like the Wellcome Sanger Institute thrives on is open access. So he means to sell the information, I guess.
Yeah, well, I can tell you to the thing that I know. In the lab that I've been, so our main focus was to be able to push the boundaries of science, to be able to discover new ways how our immune cells work. And for example, if I concentrate on my PhD, and then afterwards I will talk about the postdoc. So during my PhD, our main focus was to be able to better understand autoimmune diseases in children and what were the causes of that. So our main focus was not to be able to, let's say, not to treat, but to like generate a product of it, but just to be able to put the knowledge out there for the scientific community, so that people and like clinicians especially can better understand those very rare autoimmune diseases and see if there could be ways they could approach the disease differently. And here in the postdoc, with the type of study we do, is also like trying to push the boundaries of science to be able to do more fundamental science, but more with an idea of, if we find something interesting that can be commercialized, we would like to go ahead with it and do it. But our main focus, I would say, is to be able to understand how fundamental research, like fundamental science, is working in our CD8 T cells.
Again, the science community is just about sharing a lot of it and hoping someone else can figure out a trouble maybe you're stuck on. But thank you very much for answering that. I hope that answers your question, Jorge. And I know we've overrun a bit, so we'll end that there. That's all right. We've got thank you so much. And I found it very interesting and informative. I think I definitely learned a lot. I love the eating of the, and killing. Fantastic. But thank you so much, Ommar, for joining us today. And hope everyone has a lovely evening. So Jorge says I enjoyed too much this presentation. Very warm, interesting, and positive.
Oh, thank you, Jorge.
Very glad everyone has enjoyed it thoroughly. So thank you very much, and we'll say good night.
Well, thank you very much for inviting me, and thank you everyone for attending.
Yeah. And we've also got our last talk of this academic year on the 7th of July, which is by someone that Ommar is friends with and works with alongside.
I would recommend you attending to his presentation.
Yeah, so it's more of this plus more detail, and so make sure that you're there for the next one as well. So a bit of a teaser. So see you all soon.
See you. Bye-bye.
Bye.